Hello students, welcome once again to my class, and this will be continuation of the last chapter, that is the chapter 8, how do organisms reproduce, in the CBSE book recommended for the CBSE students. And I feel that from my teaching experience I've seen that in addition to all the teachings of the lectures and explanation that you get from your school, from other teachers, so you need to go back and refer to your own book and learn from there, or memorize those important parts. Even after understanding those content, because remembering, is difficult when you have so much to study. So your book is the best support, and that is why I'm referring to the book, the chapter 8, and for that matter, any other book that you are consulting, you are referring, you have to go line by line and word by word so that you learn the chapter thoroughly. And with that, I start my session today again. And before going to the main modes of asexual and sexual reproduction, I will refer back to the first page of NCERT book. At the bottom, there are some mention about the proteins which are transferring the genetic material to the offspring. So if there is some change in the proteins, that is DNA, RNA, etc., in the coding and decoding, so naturally there will be some changes also in the properties of the or characteristics of the offsprings, and that causes variation, students. So this I just thought to mention because it is also very important. And then coming to the asexual mode or single parent mode of reproduction, in your book they have started with fission, and I had mentioned the fission in my last video also. That is precisely what is fission is division of the organism in two parts, that is binary fission, and if it is in multiple parts, it is multiple fission. So in your book, in this page, in the page number 129, amoeba and bacteria have been referred for, have been given as an example for binary fission. And there is an important part at the bottom, that is some organisms have a typical or uncommon, rather uncommon structure, like Leishmania donavini, which is the organism which causes Kalajar. At the end, there is a whip-like structure. So they, when binary fission is taking place, it takes place oriented to their structure, according to their structure. So this is a little variation from the common modes of binary fission. So this also you have to remember. The example is Leishmania donavini. And in multiple fission, in plasmodium multiple fission takes place. Many parts, split into many parts, and several organisms are also formed. So this is about the fission, students. And then in your book, regeneration has been maintained. No, fragmentation has been mentioned. So fragmentation is again the fragments or the parts of the filament-like body becomes reproductive and comes out, and new offsprings or new plant develop. So that is fragmentation. And fragmentation example is commonly given for Spirogyra. So it has been mentioned in your book. But fragmentation is, this also has been mentioned that fragmentation is not commonly possible in multicellular organisms because they are highly developed and there are several tissues and organs. So just separating a filament or a part of the body and producing new organisms are not possible in those multicellular, highly developed organisms. So it can take place in simple organisms only, like Spirogyra. So this is about the fragmentation, students, and in your book, then regeneration is there. When a part of the organism is lost or cut, it develops into a new organism, right? Like here, planaria has been given as an example, and the steps, there are organized steps for this to happen, and the hydra is the common example, and some lines are very important which are given here regarding regeneration. However, regeneration is not the same as reproduction, since most organisms would not normally depend on being cut up to be able to reproduce. So it is not truly as a method for reproduction, but regeneration is also important mode which is studied in this chapter. Hydra and planaria are the two examples where this happens. And student, I will like to mention that this figure that has been given here, 8.3, regeneration in planaria, you have to draw and you have to memorize this, because questions like this, actually this particular question also comes giving this figure in the question paper, and you have to identify and you have to name the process and the organism where it is taking place. So you have to memorize this also. Then budding is there in our book. Budding, small buds come out, protrude out, and they are then separated and give rise to new organisms. Like as is been given here, organisms such as hydra use regenerative cells for reproduction in the process of budding. In hydra, a bud develops as an outgrowth. A bud develops as an outgrowth due to repeated cell division. It's all because of cell division. All these asexual methods are happening only because of cell division. Due to repeated cell division at one specific site. So at one specific site, repeated cell division will happen, and that these buds develops into tiny individuals. And when fully mature, they will develop into tiny individuals. Detach from the parent body and become new independent individuals. And then after they grow in tiny individuals, they get detached and grow into a separate individual, independent individual. Then in your book, vegetative propagation is there. The students, you all have heard that in vegetative propagation, some parts of the plant become vegetative and they start reproducing. They start forming new individuals, new plants. So this is commonly used in agriculture by grafting, layering, etc. So this has been mentioned in your book, and those are very important line. Plants raised by vegetative propagation can bear flowers and fruits earlier than those produced from seeds. So this is an advantage of vegetative propagation. The yield is obtained little faster. Such methods also make possible the propagation of plants such as banana, orange, rose and jasmine. This propagation of these plants are generally done by vegetative propagation. Another advantage of vegetative propagation is that all plants produced are genetically similar enough to the parent plant to have all its characteristics. So these are the very important lines, student. You cannot ignore, because questions come related to these lines in your board exam. Then we come to the spore formation, student. This is also a asexual method. Spores are formed and they are spread and then they germinate. They give rise to new individuals. So in your book, lines are there regarding this. I read some of them because it is not possible for me to read the entire chapter, but some important lines I will definitely read to help you all. Even in many simple multicellular organisms, specific reproductive parts can be identified. The thread-like structures that develop on the braid above are the hyphae of the braid mold. They are not reproductive parts. On the other hand, the tiny blob on a stick structure are involved in reproduction. The blobs are sporangia. So these sporangia spores are contained, and when they ripe, they come out, they burst, and they come out as happens in rhizopas. So it is common in fungi and also in some bacteria. The spores are covered by thick walls that protect them until they come into contact with another moist surface and can begin to grow. So spore formation is about spore formation, it is written there, and they can wait, they can tolerate, and they can wait for the favorable conditions, and when the favorable conditions are there, they germinate into new individuals. So this is the spore formation. Figure 8.6 in a book is explaining spore formation in rhizopas, how the sporangia is bursting and spores are coming out. So this is about spore formation, students, and there is another method which is tissue culture. Some cells and tissues are cultured from plant, and then seedlings or small plants are found, and they are then grown in a separate place. So here the lines are there. I am reading for them for you. In tissue culture, new plants are grown by removing tissue or separating cells from the growing tip of a plant. The cells then placed in an artificial medium where they divide rapidly to form a small group of cells or callus. This is called callus. The questions also come related to this word, that is callus. The callus is transferred to another medium containing hormones for growth and differentiation. The plantlets are then placed in the soil so that they can grow into mature plants. Using tissue culture, many plants can be grown from one parent in disease-free conditions. This technique is commonly used for ornamental plants. So these are the important information which you have to remember. So this is all about the asexual reproductive methods in your book, students. And then we come to the sexual reproduction, where two parents are involved, male and female gametes are involved. So fusion of the male and female gametes result into fertilization, then zygote is formed and new offsprings or individuals are developed. So that is the sexual reproduction, and the benefit of sexual reproduction is that, as in asexual reproduction, the genetic characters are being copied or being transferred by DNA copying from one parent and its generations, from its own generation. So variation will be there, but when there are two parents, so two generations are being copied, the DNA are being copied, and errors will be also there. So the variation will be more in sexual reproduction, and chances of developing new species will also be there in sexual reproduction. So in asexual and sexual reproduction, this is the difference. The variety of characters, the genetic characters are being transferred by the proteins, and there it is also carrying the earlier generations characters. But if there are two individuals, this is happening for both of them. So the chances of variation, the scope of variation is more. And these lines have been mentioned there in your book also. So you have to read them and you have to understand them. The gist is this only. And some important lines I will definitely I will try to read. One solution that many multicellular organisms have found for the problem mentioned above is to have special lineages of cells and specialized organs in which only half the number of chromosomes and half the amount of DNA as compared to non-reproductive body cells. So what is this problem? The problem is that if two parents are there and if both the DNA is carrying genetic characters, then n into n will be 2n and 2n into 2n will be 4n and 4n into 4n will be 16n. So if it goes on multiplying, then the numbers of these chromosomes carrying DNA, where they will go? They will go beyond any limit. So what happens is reduction division takes place, which is called meiosis. So from diploid, n and n is 2n, so 2n is diploid, and this diploid chromosome, and then 2n again reduces to n and n, is reduced to n and n, that is haploid. So this reduction division takes place, and this is the solution of that problem. So this has been written here, you have to study carefully, and in my next video I will talk about the sexual reproduction in plants, and also will start sexual reproduction in animals. So till then, wish you very best, students. Thank you so much.